We're going to do a quick little lecture on the microscope because the world of biology is microscopic. We're studying life here, right? And as we just learned, life is cellular. So when you think of a living organism, you should think of a cell. And then some organisms are made of many cells, like us, we're made of trillions of cells, and some organisms are only made of one cell. But when it comes down to it, everything alive is made of cells. And in order to see a cell, you need a microscope. Can't see it with your naked eye. So we're going to do a quick lesson here on the microscope. So there are several kinds of microscopes, but we are going to talk about the compound microscope. That is the kind of microscope you would use in a biology class. And to compound means to add to something, right? So a compound is when you add things together. And a compound microscope is very simply when you have multiple lenses together in order to magnify an object. So if you take a look down here, this shows a compound microscope. What ends up happening is light gets passed through a specimen. So if you want to look at something under a microscope, the most important thing is it has to be thin enough that light can pass through it. So if I were to take my finger and put it on a microscope, it wouldn't work because my finger is too thick to have light pass through it. I would have to take a thin sample of my skin, put it on the microscope, and take a look at it. So what ends up happening is light comes up through the specimen goes here, and then it goes through two lenses. The first lens is going to be the objective lens, and then you have the eyepiece. So when you're looking through it, the eyepiece magnifies it once, and then the objective lens magnifies it again. So you're basically doubling the magnification. So if the eyepiece is 10 times and the objective lens is 10 times, you've just multiplied that and magnified it 100 times. You just multiply the numbers together. And I'll talk more about that in a minute. So let's talk about the parts of a microscope. You need to be familiar with this. So a microscope is a pretty simple machine, essentially. Its job is to make things look bigger. And there's a couple things you need in order for that to happen. First off, in order to see anything, you need light, right? If you were to take all the lights and turn them off in your room right now, you wouldn't see anything. It's not because they're not there anymore. It's just that you need light to see something. The way sight works is light bounces off of an object. It reflects into your eye. It goes into your eye. You have a retina on the back of your eye, and then it makes an image in your brain, and you see it, right? So our brain creates the image. So what happens here is light gets sent from a light source, so it could be usually electrical. That light source gets passed up through this little opening here, which is called the diaphragm. And what the diaphragm does is it controls the amount of light. So sometimes you want a little less light, sometimes you want a little more light. That is the diaphragm that does that. The specimen sits right here on the stage, and then it travels up through the specimen and then into what's called the objective lens. And microscopes usually have at least 3. Some have 4. And each objective lens is going to be a higher magnification. So you can increase how much something's magnified. And then it passes through the objective lens and then up through the eyepiece where it gets magnified again, and then you see it, right? So that's the basic structure of a microscope. You have your light, you have your diaphragm to control the light, you have your 2 lenses, the objective lens and the lens in the eyepiece, and then finally we have our focus knobs. And we have 2 types of focus knobs. We have a fine adjustment knob and a coarse adjustment knob. And I'm going to talk more about that in a little bit. But those are the basic parts of a microscope. Now let's talk a little bit more about magnification. So the objective lenses are those lenses that the light first goes through. And as I said, there's normally 3 on a microscope, sometimes 4. And they magnify the object by a certain amount. So, for example, if you take a look here, how do you find the total magnification of a microscope? Well, it's very simple. It's always going to be 2 lenses. The eyepiece lens, which is always 10 times. So the eyepiece is always 10 times. So if I were to take the eyepiece off of a microscope and just look into it, it would make everything that I look at 10 times bigger. But that's not big enough to see a cell. So we want to magnify it even more. That's when we add the objective lens. So there's 3 objective lenses. Usually the lowest one's called low or scanning power, and then you have high power, and then you have what's called the highest power, which is sometimes called the oil immersion lens. And we're not going to really talk about that too much, but that magnifies it the most. So the objective lens, very simply, is going to be either 10 times, 40 times, or sometimes even 100 times. So what's the total magnification? You take the eyepiece and multiply it by the objective lens. So when I have this microscope on low power, it will be 10 times for the eyepiece, 10 times for the objective lens, so 10 times 10 is 100. So at low power, you're seeing it 100 times bigger. At high power, in this example, it would be 10 times 43, or 430 times bigger. And then if you really have something small that you want to look at, you would do 10 times, because the eyepiece is always 10. And then the highest power is 97 times. So this is going to multiply it 970 times. So the objective lenses are going to multiply the amount of magnification by the amount of magnification in that lens. We're going to keep moving here. So what if you want to look in the microscope? Well, one thing you'll find is when you use a microscope, it's not in focus, right? You have to focus it. And the way you focus it is by turning these knobs. And there's two knobs. There's coarse adjustment and there's fine adjustment. The coarse adjustment knob makes big movements, so coarse means large, right, or rough. So the coarse adjustment knob is going to be what you use first. So when you always start in low power. When you're in low power, you're going to use the coarse adjustment knob, and then when you get it in focus, at that point, you're going to stop using the coarse adjustment knob, and you're going to use the fine adjustment knob to kind of get it in that last little bit in focus. So coarse adjustment is the initial, and then fine adjustment is just to kind of fine tune it so you can really see what's under the microscope. So you only use the coarse adjustment knob in low power, because if you use it in high power, you can actually damage the slide, and also you will probably be doing too much, focusing. You'll lose track of what you're looking at. So the coarse adjustment knob is going to be used in low power. Then once you get it in focus on low power, you're then going to switch to high power, and at that point you're going to use the fine adjustment knob. Almost done here. How do we actually use the microscope? Well, it's really easy. You take a slide. So you get a slide from somewhere. In a moment, I'm going to tell you how to make a slide, but you're going to get a slide from somewhere. And on that slide is going to be a very thin specimen. It might be a cell. It might be a piece of hair. It might be a piece of plant. It could be anything, but you're going to put their specimen right in the middle of the stage, and the stage is where the slide goes. And then you're going to very simply start on low power, and you're going to slowly turn the coarse adjustment knob until your specimen comes into focus. Once you get it in focus, you're then going to switch it to the next higher power. So if you started in low power and you're seeing it at, let's say, 100 times, 10 times 10, then you're going to switch to higher power, and that might be 10 times 40, so now you're seeing it 400 times. And what that does is it magnifies it, so you're going to have to then get it back in focus. And at that point, you're going to use the fine adjustment knob, so you can get it really good in focus. Now, one thing that's important to remember is when you magnify something, you're making the field of vision smaller. And what do I mean by that? Well, if you've ever used Google Maps before, you know that if you zoom in to something, you can't see as much, right? If you're zoomed out on a Google Maps, you can see the whole entire area. But if you want to focus on your house, let's say, when you zoom in, you can't see the whole area anymore. You're just seeing your house. So when you are using the higher power magnification, you are going to see less of the slide. So that's something to think about. That's why you want to start in low power so you can look around to see what you're looking for, and then once you find it, you can switch to high power. And then finally, now we're not going to be able to do this in the virtual environment, but if we were in school right now, you would be making some slides. And what you want to do to make a slide is very simple. You take your slide and you put your specimen in the center of the slide. You then put a drop of water on the specimen, and you take something called a cover slip, which is a small piece of glass or plastic, and you're going to take it and you're going to put it at a 45-degree angle like in this picture right here, and then you're going to close it like a door. You're just going to lay it down on top of the specimen. And that's important because it pushes air bubbles out. Air bubbles are very deceiving on a microscope. They look like something really exciting, but then when you really take a look, you realize it's just an air bubble. So by pushing that slide down, that gets rid of air bubbles, and then the water holds everything together. And then you start off right where I said on the last slide. You go on to on low power, find your object, get it into focus, and then switch to high power. Thank you very much.